So welcome everybody to the CEMB Future Leaders in Mechanobiology seminar series. It's my great pleasure today to introduce our speaker, Dr. Lisa van den Broek. Dr. van den Broek is a postdoctoral researcher in the Cezanne Lab at North Carolina State University, although she's currently located in Belgium. And she did her PhD at the University of Ghent with Dirk Inze, studying drought stress and physiological responses to drought stress in plants. And her current work is a mixture of computational, mostly computational, and a little bit of experimental work, looking into the interactions between stem cell divisions and root development. So today, she's going to present a talk entitled Exchange of Molecular and Cellular Information, a hybrid model connecting regulatory interactions with stem cell divisions in the root. We're very pleased to have you, Dr. van den Broek, and the screen is yours.
Thank you. Share. Okay, awesome. Okay, so you can all see my screen?
Yes, we can.
Okay, so thank you for coming to the presentation, and thank you, Charles, for organizing and inviting me to this talk. I will talk about a manuscript that we published this year in Quantitative Plant Biology, and it's entitled A Hybrid Model That Integrates Stem Cell Divisions and Key Regulatory Networks. So during this presentation, you will see a mixture between molecular data, data analysis, and the integration of this data through computational modeling. With experimental data, lots of molecular mechanisms have been unraveled, such as signaling pathways. But as experimental data is increasingly being generated, more and more downstream analysis pipelines are being developed, such as transcriptome analysis. And to bring all this data together and make sense of the system as a whole, computational models are being developed that integrate different sources of biological data, which lead to prediction-driven hypotheses. So specifically, we are researching plant growth. The control of growth and size is difficult to address, largely because the growth of a plant or a tissue is not the result of a single discrete process, but is under the control of a great diversity of signaling mechanisms that are regulated at different scales. So ranging from the molecular scale to the cellular scale, and even to tissue, organ, and the entire organism. Additionally, there are mobile factors such as peptides and transcription factors that allow cells to communicate with each other and bridge the gap between molecular and a cellular scale. And although not my expertise, I also think that through mechanobiology, cellular signals feed back to the molecular scale by mechanical factors that elicit different signaling mechanisms. So we want to understand the control of cell growth and specifically cell division by integrating and bridging this molecular and cellular scale. So how do plants achieve the right amount of growth across these scales? So plant growth and development are very complex, as I mentioned, and dynamic, and they need a multi-scale and multidisciplinary approach to identify the different players that are involved in their regulations. And in our lab, we study plant growth on a molecular scale through, for example, transcriptomics, on a cellular scale through phenotyping and reporter lines that integrate cell specificity. And we also make the connection between the molecular and cellular scale by studying the cell-to-cell communication through scanning fluorescent correlation spectroscopy that can quantify the movement of proteins between the different cell types. The integration of all this biological data through computational models is kind of the central theme in the Cezanne lab. We started with a simpler model and have not yet integrated these mechanical factors into our model, since it's not our expertise, but I will be happy to meet with the experts in the field for potential collaborative efforts. So I think where mechanobiology greatly fits our research objective, so the study if stem cell divisions, is the study of cell division orientation, as well as because cell division orientation is of particular importance in plants where the cells cannot rearrange. So the position of new cell walls simultaneously determines the cellular topology, so the neighbors of the cell and the cell-to-cell interactions, as well as the cell geometry. So within the mechanobiology field, the computational models can aid in predicting the cell wall formation in symmetric divisions, microtubule patterns. The position of the nucleus can be predictive for asymmetric divisions. So I think that to further study plant growth, the integration of these mechanical factors into the computational models that we have currently could be very interesting. So today I will talk about the Arabidopsis root stem cell niche and proteins that are involved in regulating the stem cell divisions within that niche. And for the most part, there's a comment in the chat. Also, I think cell size control may have mechanical limits. For sure, I completely agree that cell size will also have mechanical inputs. I read a little bit before this talk more about mechanobiology. There are lots of interesting stuff going on. So I will just talk about the proteins that are involved in regulating the stem cell divisions within that root stem cell niche, and the computational models that we developed to describe the dynamic behavior. For those who are not so familiar with the Arabidopsis root stem cell niche, it's a very small region in the root tip, which have cells in a very organized and specific structure. Centrally located, you have the quiescent center, which are the blue cells on the image, flanked by the vascular initials, the columella stem cells, epidermal initials, and cortex and endodermal initials. The QC, as well as the stem cells within the niche themselves, have self-renewal capacities, but also the capacity to differentiate into specific tissue files. For example, the CEI will give rise to the endodermis and the cortex cells throughout the root. The specific organization of the root stem cell niche, so the position of the cells and their cell identity, is influenced by the local environment that includes these mobile proteins and peptides, phytohormones, and all kinds of other signalings coming from neighboring cells, including mechanical factors. So two of these proteins that we know that regulate stem cell maintenance are WOX5 and shortroot. They are both mobile factors. So they can move from one cell to another. So shortroot itself is expressed in the vasculature, but the protein moves from the vasculature to the QC, CEI, and endodermis, while WOX5 expressed in the QC will move from the QC to the columella stem cells and vasculature initials. And we also showed that WOX5 can move to the CEI with scanning FCS. We use specifically pair correlation function, which is very handy. You can identify whether or not a protein crosses a certain barrier. In our case, we checked whether the protein could cross the cell wall barrier, but you can also check if it could cross, for example, the nucleus and stuff like that. So this is more of a qualitative analysis. And these spatial restrictions and concentration gradients of these regulatory proteins, they contribute to the cell type specific expression patterns and stem cell maintenance. So we were interested in these proteins and especially in WOX5, although the role of WOX5 is already extensively described in the QC and CEI, it's not much is known about its function in regulating CEI divisions. So to identify whether WOX5 has a role in the CEI, we looked at the phenotype of a mutant, WOX5 mutant, in and quantified the percentage of divided and undivided CEIs. We found that 4- and 6-day-old roots have an increase in divided CEIs. Additionally, when we crossed the CEI marker cyclin D6 into the WOX5 background, we observed an extended expression pattern of this marker into the endodermis and cortex cells, like you can see on the image. Normally, this marker is only expressed here in the CEI cells. And the cells that have these extended expression patterns were named the CEI-like cells. So this shows that WOX5 has a functional non-cell autonomous role in the CEI. So to gain an insight into the transcriptional events regulating the extended expression pattern of cyclin D6 in the WOX5 background, we performed transcriptome analysis on FACS-sorted CEI cells. So we looked only at the CEI cells because there's so little of them that if you look at the entire root, the effect is completely diluted. So we took the cell-specific CEIs. And there were 163 differentially expressed in the CEIs in the wild type background versus the non-stem cells, 200-ish in the WOX5 CEI and CEI-like stem cells versus the non-stem cells. And we hypothesized that the regulatory genes that were underlying the cyclin D6 expression should be differentially expressed in the CEI cells of the wild type and of the WOX5 roots. So we focused on the genes that were overlapping between these two sets of DGs, which were a total of 46 genes. To identify the key regulatory proteins among these 46 genes, we predicted causal relations between the transcription factors and downstream genes and constructed a gene regulatory network using dynamic Bayesian principles, a probabilistic approach. So we also, on a side note, we also developed a user-friendly interface that allows everybody to perform its own network inference with a minimum computational knowledge. And this is published in the Plant journal. This is the publication for that. So our inferred network contained 20 nodes, and 4 of them were transcription factors. So now we wanted to know which transcription factors have the largest impact on our network. And so to identify them, we developed a method that can calculate the impact of a gene in a network based on its downstream target, but also based on its upstream regulators and the secondary targets. So these are the targets that are regulated by the direct targets. So the second layer in the hierarchy. So because you can envision that a gene that regulates genes that in turn regulate a lot of genes will have a much higher impact than just a gene that regulates end node targets. So to take this into account and not only look at the out degree of a gene, so the number of genes it regulates, we developed this R-based Shiny application, which we call the node analyzer, that calculates the weights and impacts of each gene within a network. This node analyzer could be used for any network, not only a gene regulatory network, but also, for example, protein-protein interaction network or something. So the node analyzer allowed us to rank the genes within a network. And the most impactful gene within our network was AN3, angustifolia 3. which is involved in cell proliferation during leaf and flower development. So, which was of course super cool. So we decided to further study AN3 to confirm whether it also plays a role in these CEI divisions. So it was already previously shown that AN3 was expressed in the root stem cell niche. And that same paper also showed that a triple mutant, AN3 and its closest, two closest homologs, displayed a disorganized QC and increased root length as a result of an increased root meristem size. So we confirmed the growth repressing role of AN3 in the roots with an AN3 mutant line, as well as an AN3 overexpression line. And we also confirmed the disorganized QC in the AN3 mutants and showed that the mutant contains starch granules, you can see it here, in the cells that are normally the undifferentiated columella stem cells. So they should not contain these starch granules if they are kept their right identity. So then we wanted to know, after confirming the role of AN3, whether it also plays a role in the CEI division. So we basically did the same experiments as we did for WOX5. We quantified the number of undivided and divided CEI cells. And we also crossed the AN3 mutant with our cyclin D6 marker. And we had similar results as for the WOX5. So we got this extended expression pattern for the marker, and we had an increase in divided CEIs for the AN3 mutants. It's about 25% increase. So taken together, we propose that WOX5 and AN3 regulate CEI divisions. And now to gain insight in these system level regulations of CEI stem cell divisions, we modeled the expression of cyclin D6 and its known direct and indirect upstream regulators, which was WOX5, AN3, but also short root and scarecrow, which I'll talk about in a sec. So how do all these key regulatory proteins coordinately regulate stem cell divisions? So the model that we developed was a hybrid model that combines agent-based modeling with ordinary differential equations. And we included 4 cells types or agents into the model. A QC, a CEI, of course, vasculature initial, and the endodermal cells. And we constructed ODEs for all these genes for each cell type to describe the dynamics of the upstream regulatory interactions at a molecular scale. So the cells and the agents, they interact with each other through movement of short root and WOX5. And they change their state so that when they divide upon changes in the concentration of specific proteins. So here are the listed included regulations. They are the inhibition of short root by WOX5 in the vasculature, the activation of scarecrow by the short root scarecrow complex in the endodermis, QC and CEI, the activation of scarecrow by AN3, and also the activation of cyclin D6 by scarecrow and short root complex in the CEI. So by combining these 2 model types, agent-based modeling and ODEs, we could exchange information from a molecular scale, so these regulatory interactions, and a cellular scale, which are the cell divisions, so the change of state. Okay, so this is a little bit detailed about the ODEs, but I will keep it brief. So the ordinary differential equations are composed of a production term and a degradation term. As the name gives it away, the production term determines how fast the protein is produced, while the degradation terms determines how fast the protein is degraded. Within the production term, the regulatory interactions upstream of that protein are taken into account. So for example, for the cyclin D6, you have 4 different situations. One where nothing binds to the promoter, which is given by this term. One where short root binds to the promoter, one where scarecrow binds to the promoter, and one where the complex binds to the promoter. Only when the complex binds, the cyclin D6 expression will be activated. So this term of the complex will be in the numerator, while the other terms, all the other combinations that can bind to the promoter, will be in the denominator. So it's just to show you that these ODEs, that the protein concentration will be determined by the upstream transcription factors that we include. And I also just want to show that besides these upstream regulators that we include in the ODEs, we can also include the oligomeric state of the transcriptional complexes that regulate the proteins. So, for example, besides this short root scarecrow complex, there are 2 other complexes, stoichiometry complexes that can activate the cyclin B6. But including this in the ODEs really complicates the ODE quite a bit. So for this reason, and also because the expression profiles of all these complex stoichiometries were very simple, were very similar, we decided to keep the model simple and include only the short root scarecrow complex as a combination of the 2 monomers. It's also 90% of the complex stoichiometry that is available in the CEI. Additionally, with our scanning FCS, more specifically number and brightness, we also identified the stoichiometry of WOX5 and AN3, and luckily for us, because it simplifies the model even more, WOX5 and AN3 were only present as monomers. So now we need to identify all these parameters in the ODE before we can make predictions. So for that, we want to fit the model to experimental data. And we are in luck because regarding experimental data, there are already quite some data sets available for Arabidopsis in our lab. So we use the expression of the genes in each cell type from 5-day-old roots and overlaid them onto a stem cell specific time course to obtain cell type specific expression levels every 8 hours for 4 to 6 days, for a period of 48 hours. So here you can see some examples of expression of genes in the QC. But we still had a lot of parameters to estimate, so we had to reduce the number of parameter estimation. We did a sensitivity analysis. So we needed only to test the most influential parameters. So the most influential parameters you can see here in yellow. So these we estimated, and the other ones were set to a fixed value. So this was then the ODE aspect of the model. So now we can look at the agent-based aspect of the model. So each cell, so we have 4 agents, 4 cells, has its own regulatory network based on known data and experimental data. For example, short root is only being produced in the vascular initials and not in the QC or CEI. Additionally, we also include the movement of short root to QC, CEI, and dermis, as well as the movement of WOX5, so that these agents can communicate with each other. basically, the agents or cells will only divide based on simple rules when these are fulfilled. So, for example, when cyclin D6 exceeds a certain concentration, the cell will divide. Here you can see this is the expression pattern of cyclin D6. When fitted to the data, here you can see it from OX5 and the short-root scarecrow complex in the QC. So when the expression of cyclin exceeds a certain value, your cell will divide, your CEI cell, while for the QC, when OX5 and short-root scarecrow complex are below a certain concentration, the QC will divide. So each time a cell divides, all the proteins that are in that cell, the concentration will be halved. So the protein concentrations have a direct influence on the cell division, but also vice versa. A cell division has a direct influence on the protein concentrations. So that's how we exchange information bidirectionally. So now we can run the model and make predictions. So after running the model and including these agent-based rules, the model can make predictions about the expression of the proteins and also about the timing of the divisions. For example, here we have the scarecrow concentrations that are predicted. And every time you have a dip, it's because there's a division and the concentrations are halved of these proteins. So this is yellow is the scarecrow in CEI, blue scarecrow in QC, and orange scarecrow in endodermis. And these are things that we then can test also experimentally. And so we could confirm the increased scarecrow expression in the endodermal cells. So by analyzing these reporter lines, which gives us this cell specificity resolution. And this showed us indeed this increased expression of the endodermis of scarecrow in the endodermis. I have a chat question. Why is the shape of the scarecrow in the endodermis that peak so different from the divisions in other cells? So the expression pattern of scarecrow in the endodermis is based on a measured expression pattern from transcriptome analysis from the entire root, but then overlaid with cell-specific values. So that's why we have this peak, because it's just experimentally measured like that. It does not have the dips like the CEI and the QC, because we did not include any rules for dividing of the endodermis. So we basically specifically looked at dividing of the CEI and the QC. And we added rules for that, and we followed when they were dividing, but we didn't look at the division of the endodermis, because in our case, we were interested in these stem cell divisions, so we focused on those. So these lines are experimental, not simulation. Okay, let me just go back one slide. So the question is whether these lines are experimental, not simulations. These lines are simulations, but okay. For example, here you have scarecrow expression in the QC, so you can see it has this expression pattern, and the dots are the experimentally measured measurements. For example, here you have the one of EN32, where you can also see a peak. That's probably why we also have this peak in the scarecrow expression in the endodermis, because EN3 activates scarecrow. So it's normal that the experiment pattern looks a bit like it. So we fitted our data onto these expression patterns. And so we want to look our simulations as closely as possible to the real data that we have. So these are the data that we have, the little dots. And then the lines are the simulations that we have. Yes, the parameters dictate that the peak in the endermis. I think specifically the peak for the scarecrow in the endermis also comes from its regulation by AN3. Okay, glad that it's clear. You can always interrupt. I don't mind. Okay. So besides looking at these concentrations of proteins, we can also look at stem cell division dynamics. So and specifically QC and CEI. So for the QC, we found a division at 5 days and 5 hours. So it's highlighted here with this dotted line. And that's the result between a fine balance of low short-root scarecrow complex and low expression of WOX5. And we could also see that there is an increase of QC divisions in the 5- and 6-day-old roots compared to the 4-day-old roots. The CEI divisions were predicted. We predicted 2 CEI divisions, one at 4 days and 8 hours, and one at 5 days and 5 days. What is the time here? Almost 5-day-old, almost 6-day-old plants. So there was a time of 32 hours between the 2 cell divisions. And interestingly, we could see that the CEI divisions were influenced by the QC divisions. And this is because of the change in WOX5 concentration upon QC division. So once the QC divides, so this is around this time point, highlighted with the blue dotted line, the WOX5 concentration will be halved. But WOX5 moves to the vascular initials, where short-root concentration will be increased because its repressor is decreased. Short-root then in turn moves to the CEI, so you get an increase of short-root concentration in the CEI, where it forms a complex with scarecrow, and it can activate cyclin D6. So cyclin D6 goes up as well, and that's why you see this increase in cyclin D6 here, and it reaches its point for division by the rules that we set. So the CEI divisions are temporarily correlated with the QC divisions. They have an influence on each other. So we took it then one step further, and we wanted to know if the model can capture cell divisions when genes are perturbed. So based on transcriptome data of WOX5 mutants and AN3 mutants, we calculated that there was, in the case of WOX5, 99.53% reduction, while for AN3 we had 88.12% reduction in expression. So we added this to the model, and we did the model simulation of WOX5, and we also did the model simulation of AN3 mutants. So we could see in the WOX5 mutant that we had additional CEI divisions, and between 4 and 5 days compared to the wild type, which was also the case what we saw in our phenotyping. What I showed in the beginning, there was an increase in divided CEIs. So that was nice because our model could predict this mutant line. And most likely, we think the hypothesis is that these additional divisions are the results of the removal of the WOX5 repression on short root. So you have an increase in the short root scarecrow complex, which then again leads to an increase of the cyclin D6 expression. However, with the simulations of AN3 loss of function, we get the model predicts a depletion of short root in the QC, CEI, and endodermis. And experimentally, it was shown before that the scarecrow expression is depleted in the QC in an AN3 mutant. However, scarecrow is not depleted in the CEI and the endodermis experimentally. So the regulation of cyclin D6, the indirect regulation of cyclin D6 by AN3 might not be established through scarecrow, but maybe another unknown mechanism. So this gave us like a prediction-based hypothesis that there must be something else going on besides this AN3 activation of scarecrow, which then indirectly regulates cyclin. There must be something else between AN3 and cyclin D6 that results in the phenotypes that we observed in the AN3 mutant. So our hypothesis was that there's maybe another factor, called it factor X, that can repress cyclin D6. So we changed the model, we added an unknown factor X that can be activated by AN3, specifically in the CEI. We removed the AN3 activation of scarecrow and updated the ODEs within our CEI agent accordingly. Also interesting is that during model simulations, we had to add an additional rule to ensure that there is a fixed minimum time between two CEI divisions to overcome overproliferation. And so by adding this competition between repressor that is regulated by AN3 in the CEI and the short root scarecrow complex that activates cyclin D6, the model was able to capture the cyclin D6 the CEI divisions in the wild type situation, as well as the CEI situation. So an increased number of CEI divisions, as we showed phenotypically. So now we also want to know who this factor X could be. And so to search for factor X and identify potential candidates, we performed transcriptomics on these AN3 mutant lines and overlapped them with the topless interactome, and topless is a co-repressor of transcription factors. And all the transcription factors that interact with topless are generally known as transcriptional repressors. So this left us with 4 transcription factors. You can see WRKY30, MIP7, AUXIN2, and REF1. And of these 4 repressors, so these were all differentially expressed in the AN3 mutants and were also interactors of topless. So of these 4, 2 of them, WRKY30 and MIP5, showed the highest correlation with the expression pattern that our model predicts model factor X should have. So in this case, WRKY30 and MIP7 form very good candidates to be this factor X. We haven't checked them in whether they have a phenotype, but these 2 form really good candidates. So it was cool to have this project where we made predictions with the model, we test them experimentally, we adjust the model, we check it experimentally. Okay, so with this, I want to conclude my presentation with a few take-home messages. So including interactions between agents and cells is critical to fully address system level problems and replicate these behaviors. So questions about how mobile proteins affect phenotypic changes can be addressed by instructing agents or cells to communicate with each other in the model. We found an interdependency between the CEI and the QC divisions, and we have used a multi-scale hybrid model that aims to connect our biological scales with that aims to connect across biological scales and connect on molecular dynamics with phenotypic changes. The connection between regulatory inputs and cellular behavior is very complex, and with computational models, we were able to generate these prediction-based hypotheses. And then leads me to the thank you slide. So I want to thank all the lab members, and this is my great PI, Rossani. I also want to thank Ryan, because he was a great help with this project, as well as Natalie and Adam, Adam started this project and Natalie helped me with any modeling questions. Okay, that was it from my side.
Thank you.
Thank you.
Very nice job. Wonderful talk. I'd like to open the floor for questions if people have any questions they'd like to share verbally with Dr. Van den Broek. I have a lot of questions, but I'll try to hold them.
Cool.
Maybe I'll start out with a quick question. Maybe this is more a point of clarification. So in your model, you're hypothesizing that the concentration of some of these proteins goes down every time there's a cell division.
Yeah.
And I'm wondering if you can maybe say a little bit more about what led you to that, to including that aspect in your model, given the fact that if we take a plant cell and it undergoes cell division, it's dividing in half, so the volumes of the daughter cells together should equal the volume of the parent cell, but obviously then there's cell growth, and so I'm wondering how you're capturing those dynamics in your model.
Yeah, so I looked through the literature a lot to check up on these points, because we had some questions like, is this really the case? Are the concentrations halved? But it has been not specifically in plant cells, though, in animal cells. The protein concentrations do halve upon cell division. So I'm pretty confident about it, and it's yeah.
So is the thinking there that because there's less protein production happening during cell division, like mitosis and cytokinesis, there maybe isn't a lot of new protein production that's leading to a reduction in the concentration across that cell division time?
Yeah, that is hope. Yeah, that's the possibility, and it's only okay. Yeah, I should not use. I'm sorry, this is my fault. I should not use concentration, because every time I talk about it, so we talk about FPKM values. So these are more protein abundances rather than concentrations, because when you look at the concentration, you divide it by the volume. But if you look at abundances itself, it goes into two. So I should pay attention to not use the word concentration so much.
That helps a lot in terms of clarifying what you're getting at in terms of FPKM values. And the abundance, I think, is a really interesting question too, because that suggests that there's maybe a cell size independent mechanism for controlling the regulatory activity of some of these proteins. So if we think about the genome size, right, like that's not changing necessarily across cell division, although it obviously goes up and down by 2 every time during S phase and then cell division. We can think about the relative amounts of some of these, especially transcriptional regulators, vis-a-vis the genome, and how that how those dynamics play out over time is something to think about, right? So the genome size is doubling during S phase, or the amount of DNA in the cell is doubling during S phase, and then being halved again during mitosis. And it'd be really interesting to try to map those dynamics relative to the protein concentrations that you're modeling.
Yeah, if there's no like gene dosage effects going on there, yeah, it would be super interesting. I think that cell size probably does have some kind of effect on these divisions. Yeah, how we did not include it in our model, of course, because we started with a more simple model that has our regulations that we know, and we went from there and we built on it. But as I mentioned in the beginning of the talk, it would be cool to add on to this, of course.
Great, thank you. Any other questions from the audience?
I asked all of mine during the talk.
Thank you.
It was a great talk. It was really fun to hear what you're up to, and I loved how you presented sort of the back and forth between computational and experimental. Even though we've all seen it many times, it's still always cool to hear that how people are using computation.
Yeah, and still are in all my projects. There's back and forth between computation and experimental work. We're trying to go a little bit, because these ODEs and agent-based models are super cool, but they are limited to a few number of proteins. So you cannot, because ODEs are quite heavy to estimate and to run. So you can do this with 1,000 proteins or something like that. So it's cool if you really know a small number of proteins, and if you know how they are interacting with each other, because then you can do these dynamics parts. But we're trying to look also at the bigger picture, where we can look at more the entire cell and all these proteins.
Maybe I'll ask a follow-on question to that comment, which is, in these models, is there a way to build in sort of mechanics and some of the physical attributes of, let's say, the quiescent in the context of an Arabidopsis root, or other cell types and plants? Can we capture information of the physical environment under which some of these protein dynamics are happening in a modeling context, and then have that translate back to experiments that we could design?
But yeah, there are definitely a lot of models, especially these, because a lot of agent-based models are used in these physical models, where your agents are moving, or your agents, but yeah, plant cells don't move, but for other systems where your agents move and the rules of their movement is set based on these rules. But this could be applied for these models in plants as well, right? There are certain rules that you can set up. For example, there are quite some rules known in the mechanobiology, like, okay, a cell wall should form there if these and these and these things are checked. So that is an interesting part that I think that could add. This also, what could be interesting is that these agent-based models can be done in like a physical space. So you can, which we couldn't do now, but it would be a super nice addition to the model, is if a cell divides, we basically replace the mother cell with the daughter cell. But if you have a physical space, your mother cell can exist, your daughter cell can exist, and it can like go through this space. So that would be cool to also see how these things evaluate. And I think a lot of mechanical factors can help in there. So the molecular side would determine when the cell divides, and then the physical parts would come into place, like where are the cells going, and what's the size, and what will be the neighbors.
Right, so we almost need a meta systems biology approach, right, where we're taking all of the transcriptome, metabolome, proteome information and putting that in a spatial context where people are building really nice physical, predictive physical models of things like cell division and cell growth in plants and other systems. But I think merging those is going to be a major challenge and kind of a area of computational growth. At least that's my opinion.
Yeah, same here. The tricky part will be to see how these because we included these like interactions between proteins. It's difficult to add that on a large scale.
Definitely. Challenges for the future.
Yeah, definitely super cool.
All right, any other questions from the audience? Going once, going twice. Okay, well, if not, maybe we'll thank Dr. Vanden Broek once again for a lovely seminar. And we really appreciate you taking the time to present to us today.
Thank you. Thank you for the invitation.